Good afternoon. I would like to start by thanking the organizer for giving me the opportunity to discuss a little bit with you about my previous work when I was in a postdoc in the Applied Microfluidic Systems Lab, working on mostly DNA computing and not DNA data storage. And the project was under the supervision of Anthony Genot, and at that time we were interested in CRISPR-Cas system and how could we use those CRISPR-Cas system in the framework of molecular programming and luckily we had a small introduction of Cas9 before by the first speaker, and I will just give you a little bit more context maybe. So Cas is endonuclease, so it cuts double-stranded DNA, and the particularity from the others is that to do so, it's using a cofactor, which is an RNA guide, so an RNA molecule. And this RNA molecule will dictate where the cut will happen. So it's a very nice tool for genome edition, as you probably know. And there are two requirements: the spacer, which is the target sequence that will be cut, and the PAM sequence, which is where the protein itself will anchor to the DNA strands. And at that time, some years ago, people were very interested in controlling this tool, activating it, and control the activity of Cas9 in time and space. And what most of the teams at that time were focusing on the enzyme itself. Focusing on the enzyme itself, finding orthologs or modifying the enzyme with protein engineering. And our field of work was involving more DNA and nucleic acid. So we looked at what we could use in the system, and our rationale was that for nucleic acid, a very interesting property is that if we know the sequence, well, we actually have a good idea of the secondary structure of this nucleic acid, and often the structure of the nucleic acid is more important than the sequence itself for its activity with proteins. And that was the base of our reflection. How can we alter the sequence of the RNA guide to have a reversible structure that would change upon a biological signal, giving from an off signal to an on signal first. And the first task that we had to do was to find an assay and a readout, and our assay was actually inspired for a published assay at that time, where a researcher developed this double-stranded DNA that bears a quencher and a fluorophore on each strand. And the assay is just, well, it's simply the transcription of the guide targeting the double-stranded DNA using a polymerase, and the incubation of the RNA guide with Cas9 before its injection in a medium that contains the double-stranded DNA. So we obviously use this very nice and very interesting assay, and we retrieve the data either by injecting directly the complex of Cas9 and its cognate RNA guide, or by having a one-point experiment where we would just do the transcription in the system in presence of Cas9 and the probe. And this was our main in vitro readout for all the technological development that we wanted to do. And the first thing that we thought about was how can we do an activation from an off state to an on state? So we knew that we had to mutate the sequence to keep the secondary structure intact, but to mutate the sequence so that it would be complementary to RNA or DNA trigger. And we basically looked at the literature at that time, and we screened and tested all most mutation, let's say, in different areas of the RNA guide, until we found one very interesting area between the nexus and the hairpins, which was critical for its activity. So we designed a modified guide that expressed a secondary structure that looks like a big hairpin, and actually this big hairpin has can be opened by a biological trigger. And in our case, we chose microRNAs because they were like short RNA strand, and that was exactly perfect for our proof of concept. And so the activation process would be just to have this like modified RNA guide that upon the presence of the microRNA will bind to the toehold and open the RNA guide to its active form. And the first experiment that we did is what we designed a lot of different RNA guide that would respond to only one microRNA, not another, and we assess the conditional activation in vitro. And so we performed different experiments, and we obtained this kind of matrix in which so each plot is actually a time trace of fluorescence of the fluorescent probe that we used, subjected to a combination of microRNA and modified guide. And what you can see in the matrix is that when the cognate microRNA is in presence of its guide, and only this one, you have an increase of the fluorescence, meaning that the Cas9 becomes active. And one interesting feature that at that time, we were very interested in that when we saw that, is that you seem to have some kinetics variation between different microRNAs. So the protocol is exactly the same for all the experiments, but you can see that the response is a little bit different. And at that time we assumed, and we're still assuming, that it's probably due to secondary structure of the microRNA themselves. So you have an equilibrium to take into account, and you have a different response for that. But this was a very good experimental result. We are very happy with that. And as a as a DNA nanotechnologist, if you have an activation gate, you want to build an inhibition gate right after, and that's what we did. So we wanted to gain a new function, so an inhibition from an active guide. Could we like suppress its activity? And we did the same thing. We looked back at the guide RNA sequence and checked for a structure that would not, in this case, be so detrimental for the activity. And in this particular case, we took the inspiration from the anti-CRISPR peptide that were developed at that time, that were targeting an area of Cas9 in which the upper stem here would be a little bit outside of the protein. And we said, so maybe if it's it seems a little bit outside of the protein, it's not super mandatory. So we designed a new type of gate. In this case, the upper stem is elongated and the upper stem is elongated and bigger, and it's complementary to a microRNA. So we would have an active RNA guide, and in presence of the microRNA would bind to its complementary sequence and denaturate the structure of the guide. And that's what we retrieved in the experimental part, where we incubate in the same time all components, so Cas9, the transcription tool, the artificial gene that codes for the RNA guides, and our molecular reporter, the fluorescent reporter. And we saw that in the absence of microRNA, well, the signal is what we expect from a positive control, and in the presence of the microRNA, the signal would be reduced. Well, there is still a little drift here, actually, and this drift is explainable by the fact that this is a dynamical system, and in this experiment we add a defined amount of microRNA. So after a certain while, since you produce a lot of guide RNA, you will produce more than you have microRNAs inside your system, and the system will start to work. And now that we had an activation gate and an inhibition gate, we could complexify them and merge them to have more complex logic gates. And we designed a not-AND gate, in which, in this time, you have two molecular inputs. So we choose two microRNAs, microRNA 21 and 31, and the gate, the not-AND gate, should only activate the signal if one and not the other microRNA is there. And what we showed, so we designed this kind of a double modified RNA guide in which you have an activation module and an inhibition module on them. And when we did the experiment, we actually followed our prediction. The experiment followed our prediction, where in the presence of microRNA 31 and the absence of microRNA 21 only, the system would be active. And so, complexifying a little bit more this gate, actually we shifted to another gate, which is a generalization of the not end, the XOR, in which in this time you have two guide RNAs. One guide RNA that would be triggered on by the presence of miR21, and and another guide RNA that would be triggered on with the presence of microRNA 31. And what you see in the experiment is that it behaves as we expected it to behave. In other terms, when we add microRNA 21, the signal also the activity of Cas9 is exhibited by the increase of fluorescence because of the probe cutting, or microRNA 31. And when we put both of them, actually, we have an inhibition in green, and for the same reason that a NOT gate is not stable over a long period of time, while it's still stable for at least 500 minutes. When you run out of microRNA to bind, since in our system we are producing the guide RNA in real time, the system will start. And so that's already the conclusion of my talk. And sorry for the phrasing that is a little bit confusing on this slide. We developed some RNA guides to do some engineering of the activity of Cas9, and this, in terms of molecular programming, is actually a good thing and quite useful, because we have an orthogonal system compared to a DNA system that can achieve different functions. And from a DNA data storage point of view, I'm not a specialist of DNA data storage. We had a very nice example of DNA data storage using Cas9 for random access from the first speaker, but still most of the literature uses native guide RNA. And what could we gain, and this is the discussion I would like to trigger, what could we gain by having engineered guide RNA, and what could we gain in terms of DNA data storage possibilities? With that, I would like to thank you for your attention and sensibilize you for the talk of Nicolas Lobato tomorrow, who will talk about DNA computing and molecular programming. Thank you so much.
Thank you, Alexandre. Very nice presentation. Of course, Stéphane, question.
Just curiosity, did you imagine making other gates or all possible gates? Is it feasible? Do you have some ideas about it?
Yeah, actually, so I showed you a couple of gates. We developed an AND gate also and some multiplexing function for this system. And yes, actually, that's the classical development for molecular program. You try to do as many gates as you can. And yes, that's something that we aim to do. Unfortunately, this has been first. The project has been slowed down because of the COVID first, and now it's also, like with the loss of Anthony, it's a little bit complicated. It will take some time, but we are continuing this project.
But in principle, it could be possible to use Cas9 to make all gates. That's my question.
Well, in principle, I think it should be possible. But there will be a lot of, for some part, there will be a lot of development to do. For instance, for the first gate, the activation gate, we screened manually 300 designs, something like that. So yeah.
If there is no more question, let you guys follow up later during the break, and we can switch to the next speaker. So I believe we have Usad Usmani. Perfect. Thank you again. It was very nice.